Welcome, everybody. It's Friday, June 27th, 2025. Welcome to the Florida EMS webinar. My co-host, Tanner Smita, Paul Pepe, we are very grateful to have today Dr. Nick Burgraff, who came to us via John's Vote, which was our speaker last week, who was speaking about CBD. John introduced me to Nick, and I remember I was driving on I-75, and I got on the phone with Nick. I'm completely blown away. And I've been trying to tell this story for a long time, and I've been screwing it up massively. But so Nick has, I believe, two PhDs. So we're going to call you Doctor Doctor. He's an, he's an assistant professor, as you can see, in the Department of Comparative Biosciences at the University of Wisconsin-Madison. And Nick, thank you so much for joining us. We're so excited for this presentation, and take it away, my friend.
Yeah, thanks for inviting me. Yeah, the conversation while you were driving was, it got the wheels spinning for us, so it helped all around, So it's one of those things that sometimes
Oh,
just
good.
talking it out from a different set of
Yeah.
ears is beneficial on both ends. So yeah, so I put together some stuff today, some of the work that we're doing in my lab right now at the University of Wisconsin, and so for just to give a little background for everyone, so my lab studies the neural control of autonomic and respiratory functions. So we look at a lot of brainstem neural circuits and integrate some of that with the physiology of the cardiopulmonary system. And so what we've been working on lately is a lot of stuff with opioid overdose. And so we're going to talk a little bit about the neural mechanisms of fentanyl induced respiratory compromise. And so I left it as respiratory compromise because as the story evolves, you'll see there's sort of multiple components of the compromise of the respiratory system, not just this, slowing of breathing that we tend to associate with the opioids. So, all right. So I just want to start with some take-home points. Some of these are going to be very well known by everyone here that's working with the patients in the field, but just something to keep in mind as we go through, because we're going to be diving into some of the science aspect of it and the research point, and hopefully that informs some things for clinical aspects and vice versa. So first and foremost, opioids are powerful analgesics. They're very powerful analgesics. And I don't want to necessarily straight out of the gate demonize opioids because they work very well for what they're intended for, right? They relieve pain, and that's what they do, and they do it very well. But opioids do have side effects, just like any other drug. And we do understand physiologically many of these side effects. We know that breathing slows, you get constipation, GI upset, but most of the mechanisms for these off target effects or these side effects are, as of right now, pretty poorly understood mechanistically. And so it makes it hard to really prevent them from happening. Sure, you can mask some of them, but it doesn't necessarily fix the problem. And as we're starting to uncover how these opioids work more definitively, we're finding that a lot of the side effects that are coming from the opioids are often a lot more complex than we originally assumed. And so, why do we care? Well, like I said, understanding these effects can inform what? It can inform safer use of the opioids as well as more effective countermeasures, which are both very important things when it comes to opioids and being able to use these powerful pain relievers without having to worry about the negative effects. And lastly, Peter kind of introduced this in the intro, but some of the newer work that we're doing, I'll go into that, where naloxone, we like to use naloxone because naloxone works and it works fast. But we're finding out that it's not such a benign molecule after all, and it doesn't necessarily just reverse all of the effects of the opioids. It kind of creates its own state. So we'll look into some of that stuff as well. Okay. So for just to put everybody on the same page, what are the effects of opioids? Well, opioids have an integrative effect on many different neural and physiological systems. Anywhere where there's an opioid receptor, blood vessels, brain, digestive system, lungs, liver, heart, etc. Right? But at the end of the day, the primary cause of death from an opioid overdose, as of right now, is a respiratory
Mhm.
depression. And so your respiratory rate slows to a point where you can no longer maintain oxygenation, you can no longer maintain life, and you end up succumbing to a cardiorespiratory compromise. Okay. So, what do we know about that? Well, our work in my postdoc when I was at Seattle Children's, as well as the work of many others in the field, have shown that opioids very powerfully suppress central respiratory rhythm generation. So, in the brainstem, you have this area, the ventral respiratory column, and it sends these oscillatory neural signals in order to set the pace of breathing, how fast you're going to breathe. And so we call it rhythm generation. So there's a rhythm generating center in the brain. Think of it like the pacemaker for the heart, but it's for your breathing. Okay. And whether you give fentanyl, you give morphine, whether the patients are airway intact or whether they're tracheotomized, either way, you get a very powerful suppression of breathing, sometimes up to 50%, but it's very much dose dependent, of course. So as you can see over here, you give fentanyl, you give morphine, immediately within a few minutes, breathing just starts, respiratory rhythm or respiratory rate just starts tanking. Okay. And that's very reproducible. And some of the earlier work that we were doing with the opioids was looking at how can we combat this depression of respiratory rate. And so we looked at, the ventral respiratory column and the brainstem, and it can assume different states of excitability. And so you can think of a high state of excitability, like you're running away from a bear. And if you give opioids, as expected, you don't get as much of a suppression of breathing rate. Or if the animal or the patient is about to lull to sleep, they have this low state of excitability within the brainstem, and you get this massive effect or massive depression from the opioids, right? And so what we originally wanted to do was to say, hey, can we force the network in the brainstem to be at a high state of excitability or a low state of excitability on demand so we can control how the opioids function? And you can when you do it very invasively in the lab, which is one of the advantages of the lab. Not so translational for the technique, but hey, it might inform on a mechanism. So we can microdialyze norepinephrine. We can put adrenaline directly into the ventral respiratory column, and we can preferentially increase the excitability state of those neurons. And you can completely prevent or reverse any opioid suppression from happening. Same thing as we can go after some of the molecular targets on the neurons. We can block potassium channels. We can use tetraethyl ammonium. Again, it's just another way of showing that we can force the respiratory network in the brainstem to be at different states of excitability when we give the opioid, and you can completely prevent and reverse it from happening, right? So we know for for all intents and purposes, we understand that a lot of the suppression of respiratory rate from the opioids comes from just this counterbalance of excitation and inhibition in the ventral respiratory column. And so if you counterbalance that inhibition with the excitation, you can prevent it from happening, okay? So if that's the whole story, then why are we still working on opioids? Well, the problem is that opioids don't just result in this central respiratory rhythm generation suppression. They also result in pretty profound central and peripheral effects together. And so one of the issues, especially with high doses of synthetic opioids like fentanyl, remifentanil, anything like that, is you get this tonic constriction of the chest wall. You get diaphragmatic contraction, abdominal contraction, intercostal contraction all at the same time. Sometimes we call it wooden chest syndrome. And the patient essentially locks up from the chest, right? And you also get airway collapse and airway constriction to occur. And so why is this bad? Well, what we know is that a lot of times this effect is often resistant to the effects of Narcan, and it comes on very fast. And especially in the field, when you're trying to revive a patient, when you have a full respiratory system that has no compliance anymore, it's completely rigid, you can't bag mask ventilate these patients anymore, right? You're not getting efficient ventilation to them. And so what we did is we started to look into why does this happen? Why do you get these, paradoxically excitatory mechanisms to these these muscular systems when you're giving something that should be a depressant, right? An opioid. And so what we did is we started by looking at the diaphragm. And the reason that we looked at the diaphragm first is that, in many of these scientific models, we have to use mice or we have to use rats. And when you're trying to get at some of these, finer muscles of the respiratory system, you need to be able to validate that the muscle you're recording is actually the muscle that you're looking at, because sometimes you get EMG bleed through. And so the diaphragm was the best one to do that for our model. And so what we did is we gave high doses of fentanyl. We gave 500 micrograms per kilogram. You have to keep in mind that when you're scaling doses for, rodents or laboratory animals versus humans, they metabolize it very, very, very fast. And so you have to be able to, in order to get the same blood levels of opiates as you do in humans, you have to give higher doses. So what we did is we gave the fentanyl and we got this tonic constriction of the diaphragm. Normally, so this is an EMG signal from a diaphragm. Normally you contract during inspiration. It relaxes during expiration. I don't know if you can see my mouse here, hopefully. But when you,
And We can.
You can. Okay, good. When you give the high dose of fentanyl, you get this tonic constriction of the diaphragm where it no longer is relaxing in between each breath, and you're sort of just forcing breaths on top of a tonically contracted diaphragm during expiration. So that's a problem when you're trying to efficiently breathe. And so we went in and we went into the spinal cord, and between C3 and C5, you have this cluster of motor neurons called the phrenic motor nucleus, and these are the motor neurons that directly innervate and control the diaphragm. And so what we said was we asked a simple question. Well, we have this weird excitation of the diaphragm during fentanyl overdose. So what's going on with the neurons that are actually controlling that? And oddly enough, when we went in, used electrophysiology, we recorded the neurons. Instead of shutting off like you'd expect with most neurons with fentanyl, the phrenic motor neurons actually started to fire really fast. They started to become really excited. And so that creates a problem, right? Because they now can't shut off when they need to shut off, and they start to become and create this hyperexcited state, which can help or contribute to giving this tonic constriction from happening. Okay, so we've seen that that's sort of the case, but why is this happening? We followed up. I won't bear down with any of the electrophysiology details, but we looked at some of the properties on the neurons to see what the opioid was actually doing, and it seemed to be blocking a specific channel on the neurons that was changing their activity to be a hyperexcited state. And so we did some different imaging techniques in situ hybridization to figure out what type of channels were expressed on the phrenic motor neurons. And we actually saw that there was a specific subset of channels. There's EEG and ERG channels for anyone that's interested in molecular biology. They're very well expressed in the heart as well, but they're highly enriched in the phrenic motor neurons. And what was odd is that the fentanyl molecule itself actually can sit right directly in the pore of this channel and completely block any outflow of potassium. And so this is a completely non-opioid receptor dependent targeting that the opioid sits in, completely blocks outflow of potassium through these channels and causes these neurons to start firing really fast, right? So fentanyl blocks these subset of potassium channels on the motor neurons, and it causes increased tonic contraction of the diaphragm. And the scary thing is that naloxone is not going to displace this because it's not an opioid receptor. So that once it's in there, it's sort of stuck until that opioid is metabolized. Now, so that's the chest wall rigidity. Now we're going to get on to some of the newer stuff that we're working on with airway constriction and airway closure. So like I said, the opioids, they don't just result in central respiratory rhythm generation, and they also don't just result in this tonic constriction of, or this wooden chest syndrome like phenotype that happens. You also get airway collapse and airway closure. And again, the reason why this is important is, we can reverse central respiratory rhythm generation suppression from the opioids all you want, but if that's happening on top of a closed off airway and a rigid chest wall, it doesn't do anything to resuscitate the patient, right? You can have the brain sending as many signals to take breaths as you want, but if the airways are closed off, you're not moving any air. And ultimately, we need to be able to move air in order to oxygenate and get rid of the CO2. Okay. All right. So normally here, when you're doing a recording from an animal or a human, when you take a diaphragm, so in gray here is the diaphragm EMG contracting, and blue here is airflow. And so normally every time the diaphragm contracts, you get inspiration to go into the trachea at just about the exact moment that the diaphragm contracts. And it's a very efficient system. There's not much of a delay. When you overdose on fentanyl, you start to induce these transient obstructions where the diaphragm is contracting on top of a closed off airway. And so the diaphragm's contracting. And only once it builds up enough of a negative pressure do you finally snap open the airways and you get airflow to move in very rapidly. And sometimes you can't build up enough pressure and you get complete obstructive apneas throughout the respiratory cycle. Now, we had asked a series of questions, whether this was upper airway, lower airway. And eventually we came to the conclusion that it seemed that the effects of the airway closure seemed to be primarily located down in the smooth muscle airways or the lower airways. And so what we did was we started administering salbutamol or albuterol with the fentanyl to see if we could prevent some of these airway closures from happening, because if it was a smooth muscle or lower airway related, we should be able to reverse that. And that was the case, right? So we still got the same amount of respiratory rate suppression with the albuterol or the salbutamol, but we didn't get any of those obstructive events to happen anymore, right? And so they were able to maintain more of an oxygenation, even though respiratory rate was much lower. And so This is just showing the obstructions. You give fentanyl, you get the obstructions, you give the salbutamol, and you just completely reverse that. We could do the same thing with epinephrine. It wasn't as pretty as the salbutamol, but you can do the same thing with epinephrine by giving an EpiPen with the fentanyl as well. So something that was quite interesting. But again, what this told us primarily to us that are looking at the mechanisms of overdose is that it seems to be a lower airway issue that's causing the obstructions that is leading to some of these compromised events. And so, okay, if it's a lower airway issue, then why on earth? Why are we getting airway constriction with fentanyl, right? And so the smooth muscle airways of the lower airways are very much in check by a balance between parasympathetic input from the vagus nerve and sympathetic input coming from the sympathetic chain. And so if you, just a quick background, if you activate vagal nerve signaling, you overactivate parasympathetic nervous activity, you get constriction of the airways. If you activate sympathetic activity, which is mimicked by salbutamol or albuterol and epinephrine, you get dilation of the airways, right? And so the patency of the airways is maintained by the balance between these two. And so if we had the idea then, we had the hypothesis that was, does fentanyl then enhance vagal nerve outflow to cause airway constriction? Are we getting an active recruitment of vagal nerve fibers from the fentanyl causing the constriction? In other words, is it sort of an active central event that's causing these to occur? And so, all right, we went in and we recorded from the vagus nerve directly. We recorded from specific fibers that were innervating the airways, right? And so we took specialized hook electrodes that we made that were able to actually get single nerve fibers from the vagus nerve. We could validate that it was nerve. We could, give lidocaine, and here's your EMG artifact that's going to happen with any nerve recording. But, here's your vagus nerve activity. We can use some fancy techniques to spike sort in order to directly get the fibers that are innervating the lungs. And we can look at what happens to these vagal fibers when you give fentanyl. And sure enough, when you give fentanyl, here's in purple is the vagus nerve. It doesn't shut off, that's for sure. It turns on, and it turns on pretty profoundly with the fentanyl. So here's just a little movie. Here's your brain, your vagus nerve under normal conditions. You give fentanyl, and that's what happens to vagal nerve firing. So it certainly doesn't turn off. It causes it to dance pretty profoundly. So that's somewhat of an interesting finding, right? Because now, again, you have this fentanyl that should be turning things off, and it just enhances the heck out of the nerve, which we think is causing the airway constriction. Okay, so if that's the case, why are we getting these vagal nerve fibers that are very much excited by the fentanyl? So what we can do is we can go into the brainstem, into a couple nuclei that contain all of the neurons that flow through the vagus nerve. And we can take these multi-electrode array recording probes. It just allows us to record from hundreds of neurons at the same time, 400 or 500 neurons at the same time. And we can look at how these neural networks react when you give the fentanyl. And so here's just a measurement of airway resistance, or an inverse measure of it. You give the fentanyl, this goes down, so your airway resistance goes up. And then we can look into the motor nuclei that control or have all of their axons that are sent through the vagus nerve. We can use some optogenetics to make sure that we're recording from the neurons that are innervating the airways. And you give fentanyl, and sure enough, these neurons start firing like crazy. So the fentanyl preferentially turns these motor neurons on, again, similar to the spinal cord, causing the parasympathetic outflow to be overactive and giving you that airway constriction. Why do these networks turn on? Well, we take a look at some of these neurons in these motor nuclei. We isolate them. And when you isolate them from all of their synaptic inputs, so the brain can't talk to them anymore, they actually do shut off with fentanyl. And so what does that tell us? That there is likely a disinhibition of vagal motor nuclei causing activation. So it's likely that there's an inhibitory population of neurons that's in the brainstem upstream of these vagal motor neurons that are normally inhibiting them. And then when you give fentanyl, it shuts off those inhibition signals and then releases the vagus nerve to start going overactive and causing the airway constriction. Okay, so if that's the case, if it is the vagus nerve being overactive that caused the obstructions, if we get rid of the vagus nerve signaling, which is something we can do in the lab, then we should be able to prevent that from happening. And so that's exactly what happens. We do a vagotomy, remove all vagal input to the lower airways. We give fentanyl, and with the vagotomy, you no longer get these obstructions that you normally do without the vagotomy. So it does seem that the vagal nerve signaling is what's causing these lower airway obstructions. So, all right, so fentanyl increases vagal nerve firing, which seems to be causing the obstructive events. But the $1 million question, what about naloxone? We all have naloxone. Narcan is great. It's everywhere. And it certainly does its job at, I don't want to say reversing fentanyl. I think we should probably change the wording to say naloxone is good at keeping you alive. But does it reverse fentanyl? Yeah, we'll start to see here. Okay, so why do we say this? Well, we had hundreds and hundreds of animals in the lab, patients in the clinic, and we give, I'm not directly in the clinic, but we can see some of the data with the clinic. But when you give naloxone, one of the things that was quite striking, at least to us that were, measuring respiratory activity in the lab, is that once you give naloxone, so here's a compressed ENG activity of the diaphragm, so it gives you an idea of breathing. I just took off ventilation here, so it was a little cleaner, but we also have ventilation that goes with this. When you give naloxone, you start to get these rhythmic large bursts that we call a sigh. And so a physiological sigh, for anyone that's unaware, it's an augmented breath that happens sort of on top of a normal breath. And we physiologically sigh all the time to prevent normal amounts of airway collapse or atelectasis that happens throughout the day just from gravity, pulling of blood flow, etc. And it's one of the things that needs to be programmed into ventilators to prevent the alveoli from collapsing over time. But when you give naloxone, the animals and some of the patients start to sigh really fast. They start to have these big sighs that happen very rhythmically that normally aren't there. And so here's just some of the quantified data over there. It's very much dose dependent with the fentanyl as well. And so we thought, well, what the heck? Why are we sighing? Well, one of the reasons that you do sigh normally is from an increase in airway resistance, right? You try to overcome some of that resistance, blow open some of those airways that are starting to close off. And so we asked the question, okay, well, let's just measure airway resistance, the proper way, put them on a ventilator. We want to measure size, so we can't have size in the ventilator. So when you give saline, you do get a normal amount of increase in resistance over time. But if you give fentanyl sort of speeds up that amount increase in resistance. So fentanyl definitely causes, like I said, the airway constriction of the lower airways, which causes an increase in constriction. And with every animal that we've tested so far, the interesting thing is right when you give this big dose of naloxone, or you give a normal dose of naloxone, I should say, you get this large spike in airway resistance. So the airways constrict even more than they were before. And then it does relax, but it definitely does not reverse course from the effects of the opioids here. And so you can look at compliance too. It's just basically the inverse of resistance. So it's, pretty much shows the same thing. Okay. So why is this happening? Why is the neural networks that are supposedly, sensing the changes in airway resistance having some odd effects happening from naloxone, causing you to sigh much more than you did before? And so what we did is we took these multi-electrode array recording probes again. We went into the nucleus of the solitary tract, which has most of the pulmonary afferents that are coming in, so the sensory neurons that are sensing, responsible for telling the brain, hey, we have an increase in resistance, we need to sigh. What's happening in those neurons? And we can do some different sorting techniques to find specific populations of neurons within there. So this is a couple hundred neurons measured at the same time. And what you would expect when you give fentanyl over here is you give fentanyl, you get a depression of neural activity, and you give naloxone, and then it just reverses it back to normal, right? That's what we would expect to happen from Narcan. And there certainly is a small population of neurons in this area that does that. But the odd thing here is that when you give fentanyl, some of these neurons actually start to increase their firing. But when you give naloxone, a lot of the clusters of neurons following naloxone essentially almost die off to the point where they're not even firing anymore. And you actually activate new populations of neurons from naloxone that were never active before, either with control or with fentanyl. And so it's really shifting this neural network into a completely new space. And so we can actually quantify where that neural network is at any given time. So here's just an example of a recording from the brain from these multi-electrode array recording probes. And you get all the neurons. These are the pulmonary afferent neurons that are sensing stretch in the airways. And what you can do is you can chunk it up into almost an infinite amount of configurations over time to see how these neurons of this network align themselves into specific patterns. And what you can do is you can take a snapshot of each time point within the network, and you can project it out into a 3D space so you can actually see how this neural network evolves over time. And so this is going to come at you a little bit fast, but here green is going to be control where there's no fentanyl. Once it turns red, that's when we've given fentanyl. And once it turns blue, that's when we've given naloxone. And we're going to see how this neural network evolves with the addition of the drugs. And of course, the GIF is not going to work. So normally it projects itself at you, but that's all right. That's how it works. Anyways, we'll just look at it here. Okay, green, you have this cluster. This is the neural network space of the pulmonary afferents. When there's no drug, it's very tightly clustered, just hangs out. When you give the fentanyl, you start to shoot this network into a completely different configuration. Not all unsurprising. You just gave fentanyl. It has to be able to change and adapt to the new state of the physiological system, being respiratory compromise. And when you give naloxone, it should just reverse you right back to the green and bring all the neurons back to where they were. But naloxone actually shifts you further away from control than fentanyl actually shifted you to begin with. So was a sort of an interesting finding to us, and this has happened in every animal that we've tested so far. It follows the exact same trajectory, which is quite weird. Okay, we can quantify things about it. We can look at how much time it takes for these networks to find a new stable point following the different drugs. But again, at the end of the day, the only thing that I want you to take away from this is that naloxone is not bringing you back to where you were before. It's just putting you into a new state where you can stay alive until the opioid can metabolize itself, the naloxone can metabolize itself, and you go back. Okay. So what do we know? Fentanyl causes central respiratory rhythm suppression and airway compromise, right? The chest wall stiffening is due to potassium channel block on the spinal cord motor neurons that are controlling the diaphragm. The airway compromise is in part due to overactivation of the vagal nerve activity. We know that airway compromise can be reversed with the sympathomimetics like albuterol or salbutamol. Like I said, naloxone helps prevent death, but it does not return you back to normal. So what do we know? And what are we proposing here is that combining sympathomimetics with naloxone may be a way to reduce the amount of naloxone needed so you don't stress this neural network into a new state in order to reverse the overdose and also minimize the alterations in neural network configuration. So with that, here's the people that are doing all the work in the lab, and that's what I got. Happy to take any questions.
Wow, that's pretty incredible. I mean, you've made the case here that many of us on this call have no idea how naloxone works. Fentanyl works. You've made the case that it has some negative effects to the patient, which many of us did not know. And then you have to go about reversing those negative effects with other alternatives, such as either albuterol or epinephrine. So it's all fascinating. This is incredible. Before Paul goes to the webinar, Paul, we've been bathing in molecular science that's very relevant to the clinical world. I would love to hear your thoughts before we go on to Tanner. You're on mute. Oh, you can't talk. You can't talk. Okay. All right. Tanner, you are our resident scientist as well here. You are an MD, PhD student. And so I know that you and Nick will have lots to talk about, but love to hear your comments.
Peter, can you hear me now?
Oh, I can hear you now. Yes. Go ahead, Paul.
Yeah,
Love to hear your
I
comments.
was muted and I couldn't get out of it, so I was trying. I had to re-sign in and all that. I do have to be on the other call. There was like about 15 things I could say here, and I can maybe do it next week or do it offline with him, etc. But I'll try to make 3 quick points. One is that I'm really happy about this because it's not binary thinking that something does just this. It's multidimensional in its course and has a lot of different effects, and we've known about the stiff chest wall that occurs. A lot of people think about it with other drugs, et cetera, but this effect that you're seeing here is profound, and it may contribute to the reason why people have oxygenation problems because they cannot open dependent lung zones, et cetera. And that's a whole, again, I have to go drill down on that. It's something. But one of the interesting things as well is everybody thinks it's all hypoxic mechanism of, say, a slow heart rate, but part of it is also the increased vagal tone that we see here as well. And so the concept of bypassing that vagal tone and trying to reverse it, I think is pretty profound, and it's a great finding they had here. And I always remember that, by the way, to talk about the diaphragm, right now, 80% of your breathing is done at rest here by your diaphragm. It's such an important muscle here. And so to have I think we all remember C345 keeps the diaphragm alive, and to hit those areas is really an important thing to keep in mind that that's such an important muscle, and it's also the reason why the pulmonary vascular resistance goes up when this effect happens. I mean, there's so many other complicating effects that you see here. But real quickly, because I do have to get into this other call, and we could go on to other things. I do want to know, have you seen the vascular effects as well, or are you studying those yet? Because this affects smooth muscle all the way around.
Yeah, we haven't looked at the vascular effects yet, other than the fact that when we do put a catheter in, obviously you do get the hypertension that can happen.
Right.
But that's to that extent, that's the only thing that we've done. We wanted to look at the fact that, with albuterol or salbutamol or epinephrine, you do have to worry about some of that vascular constriction being worsening.
Yeah.
And so even if you do open up the airways, you may reduce oxygenation. So there is a bit of a dose thing that has to be fleshed out. And I think
Yeah.
if this thing has legs to get into a patient population, I think that there's groups that can do it much better than we can in rodents. But yeah, it's something to
And
keep in mind.
I'm really glad you brought up the size, because people have no idea about that. It was kind of the guy who first discovered that back in the 50s, Henry Ben Dixon and his team in Mass General. They were so good at spotting size, they can spot a sigh 13 rows behind in church, that kind of under a mint coat. That they were looking at that. It was profound, because when they first started breathing people with 5 to 7 mils per kilogram, and they started seeing desaturations, it was like, oh, what's going on? And then they discovered what was happening there. The body has reflex. So I think the size effect is actually is a reaction, a compensatory reaction to the stiff diaphragms. I think that's what you were alluding to, correct?
Yeah, we are. Yeah, real quick, we have two hypotheses for the size. One is that normally fentanyl or opioids do suppress the ability to create size, and so as the resistance is going up, when you finally give the naloxone, you completely remove the fentanyl off the opioid receptors, and now you're starting to try to overcome some of the complications with the airway collapse or the constriction. So hence the size. We're testing that right now. The other hypothesis is that you have a refractory inhibition in the neural networks. Anytime you hold down a rhythmic neural network, especially the respiratory network, over time, it starts to try to compensate. So it starts to dump glutamate, substance P, all different excitatory neurotransmitters in there to overcome the effect. And when you immediately remove the inhibition from the fentanyl, now you have an overexcited network, which we know causes it to sigh to try to reconfigure itself back into a stable state.
So what you're saying to me too is that there is this more things than just one effect. It's multi-thing, and sometimes there are compensatory effects. There's, who knows what else, primary effects or whatever. And so it's very, so it's great work. Real quick, when were you at Children's in Seattle?
I was there from 2019 to 2024.
Oh, okay, got it. All right, good. So those are the days after I was there with Len Hudson and that whole crew, and Brownie Shaney and all those guys years ago, and and they were just the most brilliant people. Last thing I was going to say is that did Azita hire you, or was she gone already?
She's gone. She might be here, but I'm in the comparative bio department. So
yeah, okay, got it. So different department. Okay, good. All right, listen, we'll catch up later. But this was great, and thanks for getting this out to people, because they need to know about this. So I think that's so cool. Okay, and I got to go to my call. I'm really pretty late.
We'll be right over. Thanks, Paul.
All right,
appreciate you. All right, awesome. And then Nick, maybe after Tanner's comment, I do want you to talk about the setup you had with Seattle Fire. You were going to looking at albuterol plus fentanyl, but before you get into that, plus naloxone, sorry, but go ahead, Tanner. Let me hear your comment.
Sure. Yeah, Nick. Again, thank you for this presentation. Beautiful figures. I think a great example of science and physiology. Really enjoyable. So as kind of a clinician scientist in training, and this might kind of feed into what you're going to talk about with Seattle Fire. I just wanted to talk about some perhaps limitations in translating this out to the field and into pre-hospital medicine. So I know that, and you touched on this briefly in your presentation, but the doses of fentanyl that are being administered to these animals are a lot higher than we see in both clinical practice and likely also in human victims of drug overdose. So I believe in some of these studies it was up to 500 micrograms per kilo, whereas in clinical practice, as a bolus doses, we're giving like 1 to 2 mics per kilogram. And the lethal dose for primates, while it's really high in rats, it's like over 3 milligrams per kilogram. It's only 300 micrograms per kilogram in primates. So, I think and you can comment on this if there's any evidence out there whether kind of physiologic doses that we're seeing in human victims of overdose may produce similar effects. Um,
Yeah.
I'd say that, there's a lot of patients in our ICUs that are receiving, for example, fentanyl anesthesia, high doses for long periods of time. And while there is some human data out there to suggest that it increases airway resistance and causes bronchoconstriction, right, we're still able to oxygenate and ventilate these patients. And so building off of that, as EMS clinicians, when we're out in the field, our real concern is getting these people alive to the emergency department. And so I think a question kind of remains, is fentanyl, these kind of other effects, the rigid chest syndrome, um, the bronchoconstriction, is it significant enough that it's going to impair our ability to oxygenate and ventilate these patients? I know personally, in my experience, now I've participated in the treatment of a lot of patients with opioid overdose, and I've never been in a scenario where I've seen bronchoconstriction or seen a failure to ventilate these patients outside the context of maybe, airway catastrophe, contamination with blood, emesis, that sort of thing. Final point. So we talk about administration of sympathomimetics, albuterol, epinephrine. I know one concern that's kind of been bandied about, it's been talked about in the context of case reports and some small cohort studies, is the risk of the development of pulmonary edema following administration of naloxone. Part of the hypothesized mechanism, right? You get, laryngeal spasm, you get inspiration against a closed glottis whenever you give naloxone, and that can lead to pulmonary edema that can be, pretty devastating in some of these case reports, right? People have, acute respiratory failure, require CPAP, BiPAP. And so I'm wondering, what are your thoughts on the risk of when you're administering these sympathomimetics, you're increasing afterload? May that increase the risk of this pulmonary edema that we're all warned about? I know it's very rare on the case report level. But just interested to hear your comments on those and some thoughts toward where is this going in terms of clinical translation?
Yeah, and so a couple points on each of those points that were all there and all valid. And it's something certainly is as we're developing these scientific approaches to overcoming opioids. This is the kind of information that we need, because we can mimic certain things of overdose and we can sort of force our own symptoms of overdose. But at the end of the day, we're trying to inform for human medicine. And so whatever the most important side effects or the most important effects of the opioids that you guys are seeing are the ones that we need to focus on. When it comes to the differences in dosing, this is one of the things with trying to translate between rodents and humans. It's always been a bit of a contention. And I think the way that it's been compromised to at the point right now is that we know that rodents, with any sort of drugs that we have to give significantly higher doses just because they metabolize it so fast. And so what we've been going off of is the amount of respiratory suppression in order to mimic the effects that are seen in the humans. So we give the same dose that allows us to see the same increase in blood pressure, change in temperature, and decrease in respiratory rate that you see in the human patients. And so as of right now, that's the best that we can do is we can mimic the physiological effects with a dose that's appropriate for the animal to give that. And certainly, when we were, when I was in Milwaukee, we were working on goats instead of mice. And so the goats by contrast used much less opiate than that of the mice. And, some of that might just be the fact that the mice are already, they're prey animals. They're very, tense as it is, and so it's kind of hard to grab a mouse, give it an opioid without it being hyperadrenalized to begin with, which we've shown prevents some of the effects of the opioids. And so as of right now, that's what we have that we can best do right now is to mimic the physiological effects, which is ultimately what we're trying to overcome. As far as the rigid chest syndrome, I think, where I see this research fitting mainly is in the emergency medical situations, or the first responder type of situations. I think in the clinic, you have much better control over the effects of the opiates. You can give oxygen better. You can give a paralytic if you start to get some of these wooden chest-like effects. I know when we were talking to Peter that you guys are carrying some paralytic with you out in the field, which I think is a great thing, especially for this to be able to overcome that if you absolutely need to get either a tube in or bag mask ventilation. And so I think, what we're dealing with mainly, and I think that's one of the gaps that it's always been there for the opiates is, by the time that the patient gets to the clinic, most of the medicine for that patient is already done, right? It's mainly surveillance by the time they get to the clinic. And maybe you have to overcome a few other things. But I think where a lot of this is informing is how can we, as a first line treatment, create different adjunctive therapies in order to overcome some of the effects that are going to be from that immediate overdose. And I think that's one of the areas that's been lacking in the opioid field very significantly, because again, like you said, with the patient's anesthetized or the patient has a paralytic on board, or if they're trached, or if they're on a ventilator, a lot of these effects can be overcome, right? You can just manually force a bit more air in there. You can change the oxygenation. You can paralyze them a bit stronger. Like all of these effects can be overdone. Where they can't be overdone is when you have that patient on Third Avenue and Pine that is bent over, and we're waiting for EMS to get there because traffic is clogged up. And now they get there and they have a patient that's very well on their way, strong into the overdose. And so it becomes a question of, is there a way that instead of giving that hammer approach of jamming, as much Narcan as we can to just get them back to life, is sort of this idea that we were testing when I was in Milwaukee with COPD as well, is, you have these patients that are chronically hypercapnic. We can't just remove that. They've adapted to that. Right. And same thing, these patients have been on opioids for a long time, and now they're, overdosing with the big single bolus dose. And so do we want to just completely create that? And one of the effects, one of the negative effects of giving that hammer approach is they go into immediate withdrawal. Well, do we need to put them into immediate withdrawal? And do we need to be putting these emergency medical providers at risk when you have patients that become very agitated and angry? And I think some of the data are now pointing to the fact that we may be able to use a lot less Narcan and sort of, help push them into a state of recovery without giving the sledgehammer. And I think if you use adjunctive therapies to overcome some of the other physiological effects, I think that is possible. So that's, I guess, my stance on that. But yeah, definitely you're right. When it comes to a surgical situation or already in clinic, I think that's a totally different ballgame.
And Nick, adjunctive, you're meaning the albuterol or an epi, like an IM epi, are the two adjuncts. Yeah.
Yeah. Yep.
Lastly, I have to run too here, but David Dillon out of Northern California came out with a paper looking at Narcan and cardiac arrest and found that not only did it help folks with opioid overdose, it helped folks who did not have any opioid in their system. Can you track what you're talking about here today with Narcan, with the sigh breaths, etc., as potentially the mechanism why those folks had a better outcome, neurologic intact outcome with Narcan in cardiac arrest, or is that too big of a leap?
No, I don't think it's too big of a leap. I think, a lot of that is speculative right now, is, none of it's been published, and we're starting to get into the field of Narcan and size. And, so whenever you're measuring breathing on a patient, if you change their temperature, if you change the excitability state, you can get them to sigh a lot more than they did before. And so it seems to be a lot of it is in, it's certainly in response to airway constriction, airway collapse, atelectasis, but it also is in very much response to changing the excitability state of that neural network that controls breathing. Because when you do a big sigh, you have to think about the fact that it recruits the entire respiratory network, and it causes all of those neurons to then go into refraction at the same time. So you can restart rhythm generation in a synchronized manner. And so anytime that you increase neural entropy, so the chaos that is going on within this neural network that's all trying to sync up, anytime you increase that, it'll cause these sighs to happen. And so the reason that I bring that up is that in addition to exogenous opioids that you're giving with fentanyl or morphine or anything like that, you do have a lot of endogenous opioids, your endorphins, your enkephalins, that are very much keeping breathing in check. And one of the things that we have tested in the past is that when you block these endogenous opiates, you create a rhythmic neural network in the brainstem that's much less able to overcome any sorts of excitability. So you give a little bit of adrenaline and it just starts to run away to the point where it almost seizes. And so one of the hypotheses that we have is that a lot of your endogenous opioids, especially that are released in times of stress or in times of exercise, things like that, one of the purposes of them being dumped very strongly into these rhythm generating networks in the brainstem is to keep some of this excitation from running away in order to provide that inhibition. And so when you give Narcan, you're removing that. You remove the endogenous ability to create inhibition into a network that's very much balanced. on a certain amount of excitation and inhibition. And so when that happens, like I said, you can start to drive size. And I think, when it comes to the mechanics of why does it help in cardiac failure, things like that, or cardiac compromise, is that when you're taking these big breaths, you do change the dynamics of blood flow, especially through the lungs and through the heart. And so when you're taking these big augmented breaths, I think it can help force some of that blood out just by pure mechanical constraint. And so I think that might be a way that it could help. And I think it's a very easy thing to test. I think it's very much approved already as far as Narcan is to be given to healthy patients. And I think it's something that would be worth testing.
Yeah. Wow. Well, I'll guarantee you here that you create a lot of spinning heads here today. And that was I knew this was going to happen because I'm hearing it now for the second time. And now I think I fully understand it based on all the images. I'm going to probably have to listen to this again.
Sure.
We've had the most views, live views on LinkedIn. So the people on LinkedIn right now, that's the most ever. We have people who are still here today. So, on behalf of myself and Tanner and everyone here from Florida and beyond, thank you so much for joining us today.
Yeah, thank you.
I have to run, but how can people get in touch with you? I know you run a podcast, I believe. So tell us how we can stay in touch with the brain of Nick Burgraff.
Sure. Yeah. I do run a podcast, The Neural Network. We talk about science stuff in hopefully a digestible manner. And then if anyone wants to get in contact, it's nburgraff@wisc.edu, or you can do nburgraff@gmail.com. Either one, send me a message. Like I said, we have the lab. We're very much open to any sort of collaboration for clinical projects. If you have an idea, we can test it. We have a lot of tools. And so, and we have some smart people in the lab.
Yeah.
So yeah, if anyone wants to reach out, feel free.
All right. Excellent. Awesome. Well, thank you. Appreciate it. Tanner, thank you. And thanks for everyone. And the webinar, if you want to go join Dr. Pepe's webinar, is right there in the link. I'll probably be on in about 15 minutes.